Well documented as a botanical medicine for over 5,000 years, the discovery of endogenous cannabinoid receptors and evidence-based reports indicating the efficacy of medical cannabis for treating pain have renewed consideration of its therapeutic use. The past quarter-century's advances in healthcare science has included the discovery of a previously unknown human endocannabinoid system dedicated to maintaining critical balances between our mind and body. The following recording derives from a 6-part series of sequential lectures concerning endocannabinoid receptors and medical cannabis, as narrated by physician expert and author Dr. Jeffrey Block. This publication emphasizes how an appreciation of the biologic context to show how and why endogenous cannabinoid receptors function allows healthcare providers to better interpret the limited evidence-based data. In order to properly assess the abundance of anecdotal reports concerning cannabis, the safe and effective treatment of pain will require careful clinical monitoring of patient outcomes to establish and validate the evolving evidence-based data. Dr. Block is an American board-certified anesthesiologist and respected thought leader with a clinical scientist's understanding of the healing potential found with certain botanical medicines. Fellowship trained in both pain management and addiction medicine, his pragmatic rationale considers a citizen patient's compassionate access to the cannabis plant's bioactive chemicals when used for the treatment of pain, as well as acknowledging cannabis's obvious public health concerns ranging from contaminants to dependence. The author's insight shares contemporary perspectives by first acknowledging the bioethical obligation healers have to not simply feel compassion for their patients, but to strive to effectively treat pain. This is emphasized while at the same time acknowledging those concerns to public health surrounding addiction and accidental overdose deaths. Dr. Block's writing reveals how and why a human being's natural instinct nurtures their nature through their body's own endocannabinoids, and why a contemporary physician healer's understanding of the traditional ethnobotanical role certain plant-based medicines have played in treating the intolerable symptoms of disease appears to have also tangible value in modern healthcare. As read from Endocannabinoid Receptors and Medical Cannabis, here is the author Dr. Jeffrey Block's narration of Section 2, Plant-Derived Molecules Engage Endogenous Receptors.
The cannabis plant's bioactive chemicals' effects on animals leads to the discovery of receptors and endocannabinoids. While the endogenous cannabinoid system, or endocannabinoid system, carries the plant's name because study of cannabis effects led to the system's discovery, the bodily regulatory system is in fact exponentially older than the plant. All animals, vertebrates and even invertebrates, have an endocannabinoid system, the oldest identified being the sea squirt, a creature that developed nearly 600 million years ago. By comparison, cannabis developed as a distinct botanical genus no earlier than 34 million years ago during the Oligocene epoch, and its closest genetic relative, hops, does not appear as a species in the fossil record until only around 6 million years ago. The appearance of a botanical compound that interacts with a much older animal receptor system may support a teleological or ethnobotanical contention that there is a coevolutionary relationship in which plants have developed highly sophisticated chemistries to cooperatively appeal with creatures unwittingly assisting with those plants' reproduction and dissemination, as well as to repel insect and animal herbivore threats. An understanding of the plant's survival rationale also helps to explain why its offensively bitter-tasting defensive chemical alkaloids often are taken along with a proverbial spoonful of sugar when used as medicines to help make their ingestion more palatable. Molecular similarities attest to the core of biochemical evolutionary links between plants and animals. This becomes apparent when comparing the critical energy molecules of plants and animals. Light energy processing is possible through chlorophyll that allows photosynthesis, while animals' hemoglobin molecule is responsible for oxygen exchange to derive energy. The mere substitution of magnesium, a 2+ cation, at the core of chlorophyll with iron, another 2+ cation, dramatically changes that molecule into serving as hemoglobin. This similarity of molecular structure is most remarkable when one considers the resulting essential consequence of each molecule's unique bioactive purpose. The discovery process of identifying active plant compounds and then searching for the bodily system with which they interact has proven fruitful in many instances, although compiling the supporting evidence can often be time and cost-consuming. Aspirin's analgesic properties were commercially known for nearly a century before the discovery of prostaglandins, and of course opium's analgesic effects were known for thousands of years before endorphins and enkephalins were identified. In 1974, endorphins were discovered through plant-based phytoopioids derived from the opium poppy and morphine. In 1992, endocannabinoids were discovered through phytocannabinoids produced by the cannabis plant, and in 1997, endovanilloids were identified through phytovanilloids, including the chili pepper and capsaicin. Ethnobotanical observations of how plants affect pain have helped to identify these three distinct endogenous receptor systems, each of which has been revealed through their plant's unique chemical contributions. These three receptor systems have complementary pathways that collectively may help treat pain more safely and effectively when used in a balanced therapeutic manner. Several decades before the discovery of any of these receptors particularly important to pain perception, major pharmaceutical companies had already attempted to mitigate side effects by combining only small amounts of all three plant substances for use as an elixir. Some components of the cannabis plant, such as cannabidiol and cannabinol, were described by chemists in the 1940s, but it would take until 1964 before the definitive structure and biosynthesis of cannabis's best-known bioactive molecule, delta-9 tetrahydrocannabinol, or THC, was determined. Complementing what is known about the molecular structure of THC and CBD, elucidating these cannabinoids' biosynthesis and breakdown revealed a single-parent chemical, geranyl pyrophosphate, that derives not only the other cannabinoids, but also the plant's terpenes, chemical molecules that animals perceive as smells. Plant cannabinoids exist naturally in a non-psychoactive acid form, and each has its own unique bioactivity. Only upon exposure to air and heating does the loss of carbon dioxide by decarboxylation change the living plant's precursor acid form known as THCA into THC, the cannabis plant's clinically relevant primary psychoactive molecule. The consumption and digestion of THC, allowing subsequent liver metabolism, results in 11-hydroxy-THC, a potentially more potent psychoactive cannabinoid known to also have a considerably longer half-life than does its precursor molecule, THC. The identification of the cannabis plant's primary psychotropic phytocannabinoid enabled a search for the receptor system within which it interacts. It took nearly 30 years until THC was revealed to engage CB, or cannabinoid receptors, as a unique stereospecific isomer, fitting a natural receptor's lock and key analogy and giving its plant family name to this newly discovered endocannabinoid receptor system. This led to a far more essential discovery: the endogenous receptor activating ligand that THC mimics. Not simply one, but two endogenous ligands were discovered in the mid-1990s. Each of these endogenous cannabinoids, N-arachidonyl-ethanolamine and 2-arachidonylglycerol, can attach to either of the cannabinoid receptors, CB1 or CB2. Since then, many other minor lipid metabolites having molecular structures similar to anandamide and 2-AG have been identified that may also exert collective effects on cannabinoid receptors. For all of the importance of plant-human interactions in identifying endogenous receptors and targets for analgesia, future therapeutic applications will have less to do with the plant's chemicals and more to do with other pharmacological ways to manipulate the endocannabinoid system. This integrating system appears to be the body's key regulatory mechanism, particularly in the brain, where it is known to affect memory, pain perception, and mood, among other things. Cannabis modulates opiates as endocannabinoids modulate endorphins. Opioid medications are important tools in the treatment of pain, but their well-known central nervous system and gastrointestinal side effects are often poorly tolerated. As opioid overdosing frequency has become epidemic in the United States, their use has come under intense scrutiny. Among the proposed alternatives to opioid and other pharmacological pain treatments is medical cannabis, also known as marijuana. The emerging evidence indicates cannabinoids can exert opioid-sparing effects seen at both pharmacological and behavioral levels, although a recent Australian 4-year cohort study failed to confirm changes in opioid effectiveness. While the scarcity of clinical evidence for the medicinal use of cannabis poses a challenge for treatment decision-making, the expansive scope of chronic pain and frequency of opioid abuse suggest conventional treatments are insufficient. Clinical decisions are based on our knowledge of the risks and benefits of available treatments, including those that have yet to receive full acceptance. For all the limitations on clinical research utilizing plant-derived cannabinoids, a wealth of insight derives from understanding the supporting basic science. Appreciating the mechanism of action can inform decisions about where future benefits may lie for patient care. The evidence for the efficacy of cannabis as an analgesic includes a wealth of anecdotal patient reports, human clinical trials, experimental animal studies, and laboratory research. The preclinical findings on comparative safety and potential efficacy have thus far found limited confirmation in clinical trials for chronic pain. Studies indicating the efficacy of cannabis in the treatment of pain have been well summarized in the consensus conclusions of expert reviews conducted by the Institutes of Medicine in 1999 and again in 2017 by the National Academies of Sciences, Engineering, and Medicine. These meta-analyses of randomized clinical trials and other research studies found substantial evidence that cannabinoid medicines are effective in the treatment of pain. The National Institute's report also finds evidence of opioid substitution with cannabis use, consistent with initial survey data of medical cannabis users and statistical analyses of opioid prescription reductions in states where medical cannabis is available. However, a mid-2019 follow-up analysis revealed a reversal of that trend such that opioid overdose deaths currently appear to be increasing, indicating societal variables and regulatory effects that have been poorly understood. Before considering those findings and clinical recommendations, an understanding of what has been established about how endogenous receptors and their natural ligands function helps to illuminate why several plant-derived or phytocannabinoids, key active components in the cannabis plant, hold such promise for treating a startlingly broad range of conditions, including pain. As the 1999 Institute of Medicine report notes, basic biology indicates a role for cannabinoids in pain and control of movement, which is consistent with a possible therapeutic role in these areas. The experience of pain is a complex phenomenon involving the interaction of a number of biological systems having physiological and social aspects. Of course, pain in and of itself is not a diagnosis per se, but a symptom of many diseases and injuries. Because pain is a subjective state for which there are no reliable objective measures, caution is required in assessing the validity and reliability of data. Researching its amelioration presents a significant challenge, particularly in assessing a substance such as cannabis, which generally produces obvious psychoactive effects that make assessing a placebo difficult. Physiologically, pain processing is mediated through the central, autonomic, and peripheral nervous systems, all of which are potential targets for cannabinoids. Cannabinoids act on pain through mechanisms that are distinct from opioids, and it appears that these overlapping pathways may explain their synergistic and opioid-sparing effects. Cannabinoid receptors have been identified in peripheral, spinal, and supraspinal sites and play a clear role in central nervous system pain signaling. Brain sites involved in cannabinoid analgesia reflect the role of descending pathways of neural connections from the brain to the spinal cord. Research indicates both CB1 and CB2 receptors play independent roles in controlling peripheral pain. Pain is also one of the oldest documented reasons for which people have traditionally used cannabis therapeutically. From the role of cannabis in traditional Chinese medicine and Ayurvedic practices to its introduction in Western medicine in the 1840s. Chronic pain has the most robust scientific evidence for the effectiveness of cannabinoids in its treatment and is the qualifying condition for the vast majority of patients enrolled in state medical marijuana programs that recognize pain and the conditions for which it may be associated, constituting in some states more than 90% of registrants.
This recording has been a healthcare educational initiative read by physician-author Dr. Jeffrey Block. Dr. Block is the founder of Nurturing Nature Group Consultants, an independent and scrupulously neutral interdisciplinary consultancy group advancing endocannabinoid science through translational research within the practice of integrative medicine. To learn more, please visit nurturingnature.com.